You know, when you think about what connects everything, there's this constant sense of movement, right? A fundamental rhythm.
Mm-hmm.
Wallace Stevens, he described life as a continuing rhythmic movement of the pulse, the gait, even of the cells.
Yeah, and Nietzsche talked about that too, that sort of underlying pulse of life. It really does make you think how interconnected it all is.
Right down to the tiniest parts.
It does. And, sometimes in all that complexity, understanding something profound just hinges on noticing, well, one little point, like Sherlock Holmes said.
Ah, yes.
And that's kind of the focus we're going for in this deep dive.
Exactly, because today, that one little point for you, the learner, it's the cell, the absolute foundation of life.
Hmm.
We've gathered some really fascinating material, and our mission really is to pull out the crucial bits, the key knowledge about this incredible microscopic world, make it clear, make it engaging for you.
And it's not just, abstract science floating out there.
Yeah.
Understanding the cell has fundamentally changed medicine, changed how we see ourselves even.
Totally.
We're talking about a journey from, like, the very first time someone glimpsed this world right up to the kind of cutting-edge therapies changing lives today.
Yeah, and to really set the stage for that, picture this. Berlin, it's October 1837.
Okay.
Two lab partners, Matthias Schleiden, he was a botanist, apparently had quite the scar, and Theodor Schwann, a zoologist. Great sideburns, I hear.
Wow, all right.
They're both working under this famous physiologist, Johannes Mueller, and they're just having dinner.
Formal.
Totally informal, no notes taken, apparently. But out of that conversation, just chatting, bam, this crucial idea about the cell starts to form.
Wow, it just shows, doesn't it?
Yeah.
Big ideas can come from anywhere.
Exactly, ordinary moments.
And then, jump forward just over 20 years, 1858.
Yeah.
And Rudolf Virchow makes this really powerful statement. He says, no matter how we twist and turn, we shall eventually come back to the cell.
That really nails it, doesn't it?
It does. It just underscores how absolutely central the cell is to everything in biology, really, and disease, too.
It really does. And this isn't just like a theoretical thing. I think about my friend, Sam P. We lost him to melanoma back in 2017.
Oh, I'm sorry to hear that.
Thanks. Yeah, his story is just such a stark example of what happens when cells, well, when they go rogue. Started with just a mole,
Mm-hmm. Seemingly insignificant.
Right. But it turned into this devastating cancer. It just drives home the cell's role in disease.
It absolutely does. Sam's experience, sadly, it really highlights that downside. But, on the flip side, there are these incredible stories showing the cell's power for healing.
Right, like Emily Whitehead.
Exactly, Emily Whitehead. She was battling leukemia.
Her story's just amazing. She got CAR T-cell therapy, right, where they took her own T-cells, sort of reprogrammed them, engineered them to go after the cancer.
That's the one. They take out the T-cells, modify them in the lab, grow more, and then infuse these, supercharged cells back into her bloodstream.
But it wasn't smooth sailing initially, was it? She got really sick.
That's right. Very serious complications. High fever, organ failure. It was touch and go for a while there. Really perilous time.
Wow.
But thankfully, she pulled through. And it was the power of those engineered cells, specifically targeting and destroying the cancer cells, that ultimately led to her remission.
Incredible.
It's just a powerful example of the therapeutic potential hidden right inside our own cells.
Okay, so let's maybe get down to basics then. What is a cell, really?
Okay, so at its core, a cell is like a self-contained unit. It can divide, and crucially, it contains DNA.
The genetic code.
Exactly. And that DNA is organized into chromosomes. It holds all the codes. You can think of them like recipes to build proteins.
Right.
And proteins do most of the work in the cell.
Right.
And what's amazing is, as far as we know, DNA is it. It's the universal instruction carrier in every living cell we've ever found.
No RNA-based cells out there.
Not as the primary genetic blueprint, no. Not that we've discovered yet, anyway.
Okay, so if DNA is the instruction manual, what's the cell's part in this?
Well, the cell is the thing that actually reads the manual and brings it to life.
Okay.
Think of it like DNA is the musical score, the notes on the page.
Right.
But the cell is the orchestra, the musicians, the conductor, everything needed to actually interpret that score and make the music, the proteins.
I like that. So a gene by itself, just the DNA molecule.
It's inert, lifeless, like a book nobody reads or a score nobody plays. The cell deciphers the code, takes that information and turns it into actual physical stuff, proteins, structures, life.
That makes it really clear. And you mentioned it's a dividing unit.
Yes, absolutely fundamental. Inside the cell, you have molecules mostly proteins again, that kick off the process of popping the entire DNA blueprint.
Duplication.
Right. Then the cell's internal structure kind of reorganizes itself. The duplicated chromosomes get pulled apart, and finally the whole cell splits right down the middle.
Into two daughter cells.
Exactly. Two identical daughter cells.
Huh.
And this division, it's the engine behind, well, everything. Growth, repair, regeneration, reproduction, all the key features of life.
Mitosis and meiosis.
Right. Mitosis for making identical copies for growth and fixing things. And then meiosis, that's the specialized division for making sperm and eggs for sexual reproduction, having the chromosomes.
You know, hearing you talk about seeing cells, it takes me back, way back to 1993.
Oh, yeah.
Yeah, I was just starting out as a grad student. Elaine Townsend's lab at Oxford studying immunology.
Right.
And I remember grinding up a mouse spleen, putting the cells in a dish to stimulate the T cells. A few days later, I went to look under the microscope. It was this dimly lit room. You know, Oxford always felt like perpetual twilight.
Yeah, I know the feeling.
And there they were, these clumps, these masses of translucent sort of kidney-shaped T cells. But they had this incredible inner glow, a luminous fullness. You just knew they were alive. It was just unforgettable. Seeing that fundamental level of organization.
That sense of wonder, seeing a living cell for the first time.
Yeah, that definitely stays with you. It's a direct peek into that foundational layer of life.
Yeah.
And, you know, to really appreciate how far we've come, it helps to think about what medicine was like before the cell theory really grabbed hold.
Right. Back in the mid-1800s when Virchow was coming onto the scene, medicine was kind of two different worlds then, wasn't it?
Pretty much. Anatomy studying the body structure was actually quite advanced by then.
Okay.
By the 16th century, you had anatomists describing human structures pretty accurately. Andreas Vesalius, a Flemish scientist, was key.
He corrected Galen.
He did. He corrected a lot of Galen's older teachings, which, frankly, were often based on animals and just weren't quite right for humans. Vesalius brought this new level of accuracy to understanding our physical form.
But pathology understanding disease that was lagging behind, you called it still in a muddle. Yeah, that was a different story. Yeah, without understanding what was happening at the microscopic level, the causes of illness were murky. So early health reformers, they focused heavily on sanitation, public hygiene.
The miasma theory. Bad air.
Exactly. They thought disease was linked to miasmata, these sort of contagious fogs or bad airs that supposedly collected in unclean places. So the big push was for sewers, better ventilation.
Which probably did help, to be fair.
Oh, absolutely. There was a logic to it. Disease was worse in crowded, polluted areas, but the focus was external.
On cleanliness, the environment, rather than something internal, cellular.
Precisely. The thinking was illness must be contamination from the outside. But Virchow, as he studied medical history, he realized some earlier pioneers had seen that tissues were made of these basic units, cells.
Okay.
The big questions for him were, if cells are fundamental, what do they do? And where do they come from? In health, and importantly, in disease.
Sounds like he was really driven by wanting to know what he didn't know. You quoted him, "True knowledge is to be aware of one's ignorance."
Yeah, it's a great quote, isn't it? From his student days. Really captures his mindset. He felt those gaps in understanding keenly.
He had this restless curiosity.
He did. And he finally found his focus in medical science, specifically understanding cells. By 1847, he was pretty confident, saying, I am my own advisor. If cellular pathology didn't exist, well, he'd create it.
Bold.
Yeah, armed with his medical knowledge and historical understanding.
And of course, none of this would happen without actually being able to see this world. Which brings us to the microscope.
Absolutely crucial. Early 17th century, Hans and Zacharias Janssen, Dutch opticians, father and son.
Right.
They figured out that putting two magnifying lenses in a tube could magnify things way beyond what a single lens could do. The first compound microscopes.
Building on centuries of glass making.
Right, exactly. You need good lenses first.
And then Robert Hooke really brought the microscopic world to everyone's attention.
He certainly did.
Yeah.
His book, Micrographia, in 1665, it was a hit. Samuel Pepys, the famous diarist, called it the most ingenious book I ever read in my life.
Wow. For a science book.
Yeah. His drawings were incredible, so detailed, things people had never seen magnified like that. Fascinating, maybe a bit creepy sometimes.
Fleas, lice, a fly's eye. Those drawings are amazing, almost alien.
They really are. Apparently, he even got an ant drunk on brandy to sketch its antenna.
No way.
Yeah, but amidst all the bugs and pests, there was this one image, seemingly ordinary, a thin slice of cork.
And that's where he saw them, the first microscopical pores.
and he called them cells because they reminded him of the small rooms, the cells, in a monastery.
And that name stuck.
It did. And later, Schwann, looking at plants, recognized that great simplicity, these basic units showing up again and again.
But it wasn't like, boom, everyone immediately got the importance of the cell, right? You mentioned these valleys of silence, ideas getting ignored for a bit.
That's right. It happens.
Yeah.
Mendel's work on inheritance, for example. Groundbreaking, but pretty much ignored for decades.
Why was that, do you think?
Hard to say exactly. Maybe it just didn't fit the current thinking, or wasn't communicated widely enough. And even earlier, Benjamin Martin in 1720 suggested tuberculosis was caused by wonderfully minute living creatures.
Wow, 1720.
Yeah. But that idea didn't really take root until nearly a century later, with Koch and Pasteur linking specific microbes to specific diseases.
So it's not just about seeing something, it's about a whole conceptual shift.
Exactly. You have to move from just describing an object, a cell, to understanding its universality, how it's organized, what it does. It's a fundamental shift from thinking of living tissue as just continuous flesh to seeing it as built from these discrete autonomous units, cells, that actually connect all life.
Okay. But even as that evidence built up, there were people pushing back, weren't there? The vitalists.
Yes, the vitalists. They held on to the idea that living things couldn't just be explained by chemistry and physics.
It's a special life force.
Something like that. An organic animus. It had old roots back to Aristotle, but got tied up with Romanticism in the late 18th century. Thinkers like Marie-Françoise Xavier Bichat, they felt reducing life to just chemicals missed something essential, that spark of being alive.
A philosophical hurdle.
Definitely. Needed overcoming for the cell theory to really take hold.
And Virchow was key in pushing past that. You mentioned his cell state analogy. How did that help understand disease?
Yeah, that idea seems to have crystallized for him after the 1848 revolution. He saw the body like a state.
Okay.
The state is made of citizens,
Right.
The body, he said, is a cell state, and every cell is a citizen.
Right.
So disease, that's like a conflict of the citizens brought about by the action of external forces. It reframed disease, not some weird imbalance, but trouble at the cellular level, dysfunction in the fundamental units.
That's a powerful way to think about it, especially with the politics at the time. Problems in one part affect the whole system.
It is. And around then, Robert Koch, working in a pretty basic lab in Germany, made this huge leap. Anthrax.
Right, 1876.
He managed to isolate the anthrax bacteria, see these little rod-shaped microbes under the microscope, saw they could form dormant spores too. And crucially, he showed he could scientifically transfer the disease from one animal to another.
Systematically.
Yes, it sounds obvious now, but before 1876, that kind of controlled experiment showing disease transmission wasn't really established practice.
Wow, that really underscores the need for solid evidence. And then there's Ignaz Semmelweis, such a tragic story.
Yeah, Semmelweis.
Yeah.
He was a junior doctor at Vienna. Noticed childbed fever rates were sky high in the clinic, where doctors went straight from dissecting cadavers to examining pregnant women.
Without washing their hands.
Right. But in another clinic, run by midwives who weren't doing dissections, the rates were much lower.
So he figured something was being transmitted.
He did. Some unseen something. His solution, hand washing. Chlorine solution between patients.
And it worked.
Dramatically. Infection rates plummeted. But the medical establishment, they didn't buy it. Ridiculed him. Didn't believe in germ theory yet.
So they rejected his findings.
Strongly. They were offended by the idea that they were the cause, calling it a doctor's plague. He lost his job, faced rejection, just heartbreak.
Dude, that's awful. He was right, though.
Totally right. And eventually the data, especially with Joseph Lister's work later, proved him right.
Lister applied the germ theory to surgery.
Yes, building on that emerging understanding. He realized microbes cause wound infections. Started using carbolic acid to sterilize instruments, clean wounds.
Antiseptics.
Exactly. And like Semmelweis's hand washing, it drastically cut infections after surgery. Finally put germ theory into practice.
And then came the next big wave, antibiotics, targeting those microbial cells directly.
Absolutely. From the late 1800s to the mid-1900s, you get these amazing drugs joining hygiene and antiseptics, arsenamine for syphilis, penicillin, streptomycin for TB.
Ehrlich, Fleming, Waxman.
All those pioneers. Suddenly we had weapons to specifically kill the microbes causing disease. A massive augmentation.
Okay, so we're getting a handle on the cell as this fundamental unit, its role in disease. But the ultimate question, where did the first cells come from?
Ah, the origin question.
Yeah.
That is a deep, deep mystery.
Yeah.
Biologists guess that the earliest protocell might have needed just 3 key things. a membrane to hold it together, RNA to carry information, and some way to duplicate itself.
Like a little bubble with replicating RNA inside.
Sort of, yeah. Imagine self-copying RNA wrapped in a sphere of fat molecules. Maybe those fatty spheres could grow, absorb more fat, and eventually split, each carrying the RNA.
Has anyone tried to replicate that?
Jack Sostak's lab has done experiments showing things like that can happen. Membrane-bound spheroids growing and splitting. It's a plausible early step.
But still a step towards the modern cell.
Exactly. A very early step. DNA likely took over from RNA later as a main genetic carrier.
But you also mentioned there's still a big gap in the story.
Yes. The origin of the modern cell, with all its complex internal parts, the nucleus, mitochondria, that transition is still a major puzzle.
No intermediate forms found.
Not really. It's like the early stages didn't leave many clear fossils or living relatives. Nick Lane calls it an unexplained void, the black hole at the heart of biology.
Wow, a black hole.
We know a lot about how cells work now, but that initial jump to complexity, still largely mysterious.
Humbling, isn't it? These huge questions still out there. Okay, but let's focus on what we do know, the cell's purpose. You mentioned organism and organized share a root.
They do. From the Greek organon, a tool, an instrument. Something designed for a purpose.
So the cell is the fundamental tool.
You could say that, the tool that builds an organism. And it seems evolved for a few key jobs. One, autonomy, surviving on its own. That needs internal organization parts for energy, waste, supplies, keeping things stable inside.
Okay.
Two, reproduction, making more cells. And three, in complex organisms like us, differentiation, starting as one cell type and then specializing into all the different cells needed for tissues and organs.
And that autonomy really relies on the cell's internal structure, or those parts. Let's dig into some key ones. The membrane.
Absolutely vital for keeping things contained and stable. Back in the 1920s, Gorder and Grendel did these clever experiments with red blood cells.
What did they do?
They basically extracted all the fatty lipid molecule from the cell membranes and measured the total surface area they'd cover. And they found there were roughly twice as many lipids as needed to just make a single layer around the cell.
So it had to be a double layer.
Exactly. Their math strongly pointed to a lipid bilayer. 2 layers of lipids, back to back. Like 2 sheets of paper glued together, then made into a balloon. That's the membrane.
Creates a stable boundary. Makes sense. But how do things get across, and how does the cell talk to the outside world?
Good question. That wasn't clear from just the lipids. The big insight came in 1972 from Singer and Nicholson. The fluid mosaic model.
Fluid mosaic?
Yeah. Their idea was that proteins are embedded within that lipid bilayer. Some go all the way through.
Like channels or gates?
Exactly. They act as channels, pores, gates, allowing specific things in and out. So the membrane isn't impermeable. It's selectively porous.
Yeah.
And other proteins on the outside act as docking points, letting the cell communicate, receive signals.
So it's not passive at all. It's a very active boundary.
Highly active interface, yes.
Okay, what about inside? For a while, people thought it was just...
Protoplasm.
Yeah, after von Moll coined protoplasm in the 1840s for that jelly-like stuff inside, the view was basically a liquid-filled bag. No real structure.
But that's not right.
Not at all. It's highly organized. Inside the cytoplasm, there's this dynamic network called the cytoskeleton.
The cell's skeleton.
Pretty much. It's made of protein, filaments, actin, tubulin. It gives the cell shape, structural support, like our bones do for us.
But it's dynamic, you said, not just rigid scaffolding.
Highly dynamic. It holds organelles in place, helps the cell move, and it's absolutely essential for cell division, pulling those chromosomes apart. It's an active, adaptable framework. Okay. And then the powerhouse's mitochondria. They've got a fascinating backstory, don't they, with their own DNA?
They do. Mitochondria have their own separate genome, their own DNA. And interestingly, it looks a lot like bacterial DNA.
Supporting that endosymbiotic theory, that they were once free-living bacteria.
Exactly. Lynn Margulis championed that idea, that ancient cells engulfed these bacteria, and they ended up in this mutually beneficial relationship, became permanent residents.
Wow. Ancient lodgers turned essential partners.
Pretty much. And the fact they keep their own DNA points to that independent origin. It also means they can have their own genetic diseases and maybe be targeted specifically by therapies.
And how do they actually make energy?
2 main ways in the cell. There's a fast anaerobic way without oxygen that happens out in the main protoplasm. Breaks down glucose quickly.
But not very efficient.
No, yields only a little energy, just 2 ATP molecules. The main event happens inside the mitochondria. That's the slower aerobic process, needs oxygen.
And that makes much more energy.
Way more ATP. That's where most of the cell's energy comes from.
Got it. And there are other key players, too. Lysosomes, peroxisomes, the nucleus, all specialized, right? What do lysosomes and peroxisomes do?
Yeah, it's a busy place. The nucleus holds the main DNA, of course. Lysosomes are like the cell's recycling centers, full of enzymes to break down waste, old parts, debris.
Okay.
And peroxisomes, they're involved in detoxification, breaking down harmful molecules, metabolic byproducts. Each part has its job, its location. It's all coordinated to keep the cell autonomous, functioning.
And keeping that internal environment stable despite outside changes.
Right.
That's homeostasis.
Exactly. From Greek words meaning remaining the same. Claude Bernard described it in the 1870s. Walter Cannon refined it later. It's the cells and the whole organism's incredible ability to maintain stability inside. Yeah. Temperature, pH, salt levels, all kept in a narrow range.
So understanding all these parts and processes, it's really changed medicine, hasn't it? You mentioned moving beyond just targeting whole organs.
Yes. Think mid-20th century. Big focus on organ therapies, bypass surgery, transplants, drugs affecting whole systems. But now, knowing the detailed anatomy inside cells and how tiny malfunctions there cause disease, we're moving towards what you might call organelle-directed therapy.
Targeting the specific faulty part within the cell.
Precisely. Like gene therapy aimed specifically at fixing mitochondria in retinal cells to treat certain types of blindness, correcting the problem right at the source, the malfunctioning subcellular machinery.
That level of precision is just mind-blowing. And another absolutely core process, cell division. Mitosis and meiosis. You called it a back-and-forth game. How does that work in our life cycle?
Yeah, it's a neat way to think about it. Mitosis makes identical copies. You start with one cell, you get two just like it. That's for growth, repair, keeping tissues going.
Okay.
Meiosis is different. It only happens in the testes and ovaries. To make sperm and eggs, it halves the chromosome number.
So humans have 46 chromosomes.
Right, in our body cells. Meiosis makes sperm and eggs with just 23 each.
And then fertilization brings it back to 46.
Exactly. The zygote gets 23 from mom, 23 from dad, back to 46. Then that zygote divides by mitosis over and over to build the embryo, the whole organism.
And the cycle repeats when that organism makes its own sperm or eggs via meiosis.
That's the game. Half for reproduction, double for growth. And errors in that process, like getting the wrong number of chromosomes, can cause serious problems.
So understanding how cells divide, the controls, that's been critical everywhere in biology.
Oh, absolutely transformative. Why do cancer cells divide nonstop? How do stem cells know when to divide and when to specialize? How does an embryo develop from one cell? It all comes down to controlling cell division.
The 2001 Nobel Prize, right? Hartwell, Hunt, and Nurse.
Yes. For figuring out those key regulators,
right.
Their work impacts pretty much every corner of biology and medicine.
And one area where this really intersects with society and ethics is IVF, in vitro fertilization. Those early attempts were pretty wild, weren't they? Shuttles in 73.
Yeah, Dr. Shuttle's trying to fertilize eggs in the lab. It was definitely unconventional for the time and hugely controversial. Just manipulating human eggs and sperm outside the body raised massive ethical flags back then.
Right.
And even when IVF became successful, with Louise Brown's birth, which was a monumental achievement, the practice itself wasn't initially super connected to the deep cell biology of division cycles, chromosome checks, all that.
It was more about the procedure working.
Kind of, yeah. But now, our much deeper knowledge of the cell cycle, how eggs mature, how embryos develop, it's being fed back into IVF.
How so?
Well, researchers are targeting specific molecular checkpoints in egg maturation, for example.
Yeah.
Trying to improve embryo quality based on a detailed understanding of cell division, timing, and control, making IVF safer and more successful.
So fundamental science feeding directly back into the clinic.
Cool.
And this ability to handle embryos also led to genetic testing before implantation. Edwards and Gardner with rabbits.
That's right, 1968. They figured out how to carefully remove a few cells from a very early rabbit embryo, a blastocyst, without harming it.
And test those cells.
Yeah. They analyzed the chromosomes in those few cells and could tell the sex of the embryo. That was the proof of principle. You could do genetic assessment on an embryo before putting it back. Paved the way for PGD in humans.
And now fast forward, genome editing. CRISPR. That takes manipulation to a whole different level.
Oh, absolutely. CRISPR, adapted from bacteria by folks like Doudna, Charpentier, Zhang, Church. It lets us make incredibly precise cuts and changes in the DNA sequence. In living cells, even human ones.
It's like molecular scissors for the genome library.
That's a great analogy. Find the exact word or sentence you want in that huge DNA library and change it. Rewrite the gene. It's a profound shift in our power over the code of life. Huge potential for treating genetic diseases.
But huge ethical questions too, right? Like the JK case with the gene-edited baby.
Exactly. That case brought the ethical dilemmas into sharp focus, making permanent, heritable changes to human DNA. It forces us to have some very serious conversations about where the lines are.
Definitely. Okay, let's shift focus slightly from single cells to us. Multicellularity. How did that happen? You called it a minor major transition. What's that mean?
Yeah, that term from Grossberg and Strathman.
Hmm.
It's minor because, in a way, it's simple.
Yeah.
Just cells sticking together after dividing instead of floating off.
Okay. Seems minor.
But it's major because it happened independently so many times in evolution. Plants, animals, fungi, algae, they all figured out multicellularity separately.
Suggesting it gave a big advantage.
Huge advantage. Paved the way for complexity, specialization, all the diversity we see.
Yeah.
Cooperation between cells was a game changer.
And we can even see hints of how it might have started, like that snowflake yeast experiment.
Yeah, Ratcliffe's work is super cool. They took single-celled yeast, and just by selecting for clumps that settled faster in liquid.
Simple gravity selection.
Right. In just a short time, the yeast evolved to form stable multicellular snowflakes, and they even started specializing. Some cells underwent programmed cell death to create weak points so the cluster could break apart and disperse.
Wow. Evolution in a test tube, showing how multicellularity could arise from basic pressures.
Exactly. And how cooperation and even programmed death could evolve.
Now, thinking about how we develop these complex bodies, it wasn't always thought of as cells dividing and specializing, was it? Preformation theory.
Right. For a long time, the idea was that a tiny, fully formed miniature human, a homunculus, was inside the sperm or egg and just needed to grow bigger.
Like Russian dolls.
Kinda, yeah.
Hmm?
But Caspar Friedrich Wolff, back in 1759, challenged that. He looked closely at developing embryos, birds, and animals.
Under the microscope.
And he saw continuous, gradual changes. Organs and tissues forming step by step from simpler stuff, not just unfolding a pre-made form.
Epigenesis, not preformation.
Exactly. And then Feynman and Mangold's organizer experiment really showed how cells talk to each other during development.
The two-headed tadpole one?
That's the famous one. They transplanted a small piece of tissue, the organizer, from one frog embryo to another, and it induced a whole secondary head and nervous system in the host embryo.
Showing that cells release signals that tell their neighbors what to become.
Precisely. Cell-to-cell communication orchestrating development.
Yeah.
An early glimpse into that cellular talk.
You know, thinking about all this, IVF, gene editing, understanding development, it makes you reflect on tampering with these basic processes.
It really does. Things like IVF went from being radical and controversial to pretty standard medical practice.
Yeah.
Now we're looking at editing genes in embryos, doing surgery on fetuses in the womb. It forces us to ask, what's natural? What should we be doing? What are the long-term consequences? Where do we draw lines?
Big questions. It feels like we've learned so much about the individual cell, but it's how they connect, how the cell acts as a nexus, that unlocks the real complexity.
Yeah, O'Malley and Mueller-Will's idea of the cell as this nexus.
Yeah.
It really resonates. It connects everything. We've touched on its discovery, microbes, division, IVF, embryo stuff.
So many threads lead back to the cell.
They do. And the story's definitely not over.
And a huge area we haven't fully dived into yet is blood and the immune system, our defense force. When I think about that, my mind goes straight to my patient, Greta. She had myelodysplastic syndrome, MDS.
MDS, where the bone marrow struggles to make healthy blood cells.
Exactly. And it eventually progressed to leukemia. Her experience just hammered home how absolutely vital normal blood cell function is.
It's a tragic illustration. When those fundamental cellular processes in the blood go wrong, the consequences are profound. And just understanding blood itself, that history is fascinating, especially early transfusions.
Right, those were often disasters, weren't they? Hit or miss. Why did some work and others kill people?
Total mystery for a long time. That's where Karl Landsteiner's work, right around 1900, was so crucial. He approached it systematically.
No more guesswork.
Right. No more trying animal blood, which was often fatal. He just carefully tested mixing blood from different people, and he discovered the A, B, O blood groups.
Finally explaining compatibility.
Exactly. Why some transfusions worked and others caused fatal reactions. Revolutionary.
And the real test came in the World Wars, didn't it? Saving soldiers on the battlefield.
Absolutely. Guys like Bruce and Oswald Robertson pioneered using transfusions on a massive scale for shock and blood loss in WWI-2. Developing blood plasma storage was also huge, more stable, could be used more universally in emergencies.
I remember this one patient, massive hemorrhage, Just frantic activity, bags and bags of blood going in, watching them fade and then slowly stabilize. You really feel the importance of blood in those
Oh,
moments.
definitely. Those experiences make the science incredibly real. And it's not just about emergencies. Think about heart disease, LDL cholesterol,
Right. Statins like Lipitor have been standard for
or
years.
years, lowering LDL to prevent plaque buildup. But now, people are
Mhm.
working on potentially permanent fixes. Gene editing approaches.
Like Verve Therapeutics, Sek Kathirson's company. You knew him.
Yeah, we were at Mass General together. Sake's work, driven partly by losing his brother suddenly to a heart attack, has been amazing at finding genes linked to heart attack risk.
And now Verve is trying to.
They're developing ways to use gene editing, probably in liver cells, to permanently lower LDL cholesterol. The idea is maybe a single treatment instead of daily pills for life.
Wow. From pills to a one-time cellular fix. Okay, so back to blood components. Beyond red cells carrying oxygen, we have white blood cells, the immune system. Even early on, doctors noticed they seemed important, right? Addison seeing them in lung tubercles?
Yes, Addison saw white blood cells accumulating where there was inflammation, suggesting they were being called to the scene to fight something. But it was Eli Mechnikov who really showed what they were doing.
Phagocytosis, cell eating.
Exactly. His discovery that some immune cells literally engulf and digest invaders like bacteria was groundbreaking. He was a bit of an outsider, apparently clashed with the establishment, but his observations are key.
And that's the innate immune system. Neutrophils, macrophages, the first responders. With pattern recognition receptors. What kind of pattern?
It's clever. These innate cells recognize common molecular structures found on many different microbes, bits of bacterial cell walls, certain types of RNA or DNA or signals released by our own damaged cells.
So general danger signals, not super specific.
Right. Broad molecular signatures. It reflects this long evolutionary arms race, this dance with microbes. Our immune system learned to spot common microbial features for a quick initial response, like a general alarm system.
Got it. A general alarm. And then there's the adaptive system, the specific one, with memory. Early immunization hinted at that, right? Like variolation for smallpox.
Absolutely. Practices like the Indian Tika or Chinese variolation, deliberately exposing people to small amounts of smallpox material, showed the body could develop long-lasting resistance after a first encounter. Risky, for sure.
Yeah.
But it demonstrated that memory concept. Lady Mary Wortley Montagu saw it in Turkey and pushed for it in England.
And then Jenner and cowpox. Safer, much more effective.
Right. Jenner's genius observation in 1796, milkmaids getting mild cowpox didn't get deadly smallpox led to vaccination. Probably one of the biggest medical breakthroughs ever.
But the connection between innate and adaptive immunity, it's more complex than just first and second response, isn't it?
Oh, totally. That's a
Yeah.
core puzzle in immunology.
Yeah.
Turns out, if you knock out the innate system, the adaptive system B cells, T cells doesn't work properly either.
Really?
Yeah. Mice without good innate immunity respond poorly to vaccines. Humans with genetic defects there are often severely immunocompromised. The initial innate response is crucial for kickstarting and shaping the adaptive one.
So the general alarm actually helps direct the specific forces.
Huh. And Paul Ehrlich had early ideas about how that specificity might work. Sidechain theory.
Yeah, Ehrlich's sidechain theory was an early attempt. He imagined cells had lots of different receptors, side chains that could grab specific antigens. Not quite right in the details, but it got the ball rolling on specificity.
And the actual antibody structure came later. Bedelman and Porter. The Y shape. Right. They figured out the Y shape with the two arms, the Fab regions that bind the antigen, and the stem, the FSC region, that interacts with other immune cells, like macrophages, to trigger destruction.
And there was a big debate about how antibodies get so specific for millions of different things. Instructional versus selective?
Yes. Pauling thought maybe the antigen instructed the antibody, molded its shape, but that didn't fit with genetics. Protein shapes are coded by genes.
So Lederberg's selective theory won out.
It did. The idea is the body already has B cells making a huge variety of antibodies. The antigen doesn't instruct, it selects. It bumps into the B cell that happens to make the antibody that fits it.
Like finding the right key for a lock that already exists.
Exactly. I think giraffe neck's evolution selects for giraffes born with longer necks. It doesn't make individual giraffes stretch theirs. The immune system selects the B cells that already have the right antibody.
And that selection triggers clonal selection. The chosen B cell multiplies.
Precisely. The B cell that binds the antigen gets activated. Then it divides like crazy, making clones of itself. These clones mature into plasma cells, antibody factories, pumping out huge amounts of that specific antibody.
And some become memory cells for next time.
Yep. Some clones stick around long-term as memory B cells, ready for a rapid response if the same invader shows up again. As Lewis Thomas said, that new cluster of cells is a memory no less.
Wow. And understanding this, let us make specific antibodies as therapies, monoclonal antibodies, MOABs.
Exactly. Once we learned how to make large amounts of a single specific antibody in the lab, it opened up huge therapeutic possibilities, like the story of WH with follicular lymphoma.
Treated with an antibody against CD20 on the cancer cells.
Right. Likely rituximab, a prime example of targeting a specific molecule on a diseased cell with a custom-made antibody. Hugely successful field.
Okay, so that's B cells and antibodies. What about T cells, the other arm? You mentioned the thymus was a mystery organ for ages.
Yeah, the thymus gland. Sits above the heart. Galen saw it shrinks with age. People thought it was vestigial, functionless. Removing it from adult animals didn't seem to do much.
Until Jacques Miller in 1961.
Exactly. Miller showed the thymus is absolutely essential for the development of T cells. Suddenly, this forgotten organ was center stage in immunology.
And T cells, especially killer T cells, have that amazing ability to spot infected cells. But how? The inside-outside problem.
That was a big puzzle. How does a T cell see a virus hiding inside another cell? Elaine Townsend had a key insight. He showed that fragments of internal viral proteins, like the NP protein from flu virus, could somehow get displayed on the outside of the infected cell.
Okay, but how? Zinkernagel and Doherty figured that out. MHC class I.
Yes, their Nobel Prize-winning work.
Yeah.
They discovered killer T cells don't see the whole virus protein. They see small fragments, peptides, presented on the cell surface by a special molecule, MHC class I.
So MHC class I is like a display stand.
Kind of. It's constantly sampling proteins from inside the cell. Self-proteins, viral proteins, whatever, chopping them into peptides and holding them up on the cell surface. Like a molecular spy sending signals about the cell's internal state.
A peptide bearer.
Right. And for a killer T cell to attack, it needs to recognize both the self-MHC molecule and the non-self peptide it's presenting. Dual recognition.
That's incredibly specific. And the structure of MHC fits this job perfectly.
Amazingly so. When Pam Bjorkman solved the crystal structure, you could see exactly how it was shaped to bind and present those peptides. Another beautiful example of biological engineering at the molecular level, like a sperm's tail being perfectly designed for swimming.
And understanding T cells became incredibly urgent with HIV/AIDS. Finding the virus itself was the first step.
Absolutely. Montagnier and Barré-Sinoussi first identified the retrovirus in '83. Gallo confirmed it as the cause of AIDS in '84, HIV.
I remember your story about the patient with weight loss, the difficulty getting the history, the denial.
Yeah, that was tough. The patient just wouldn't acknowledge any risk factors. It highlighted the stigma, the difficulty sometimes in just getting the facts. Reminded me of that Rushdie line about examining a patient through a white sheet. You can't see clearly.
But thankfully, treatments came along, antiretrovirals.
Huge progress. Turned HIV from a death sentence into a manageable chronic condition for many. And prevention strategies like preventing mother-to-child transmission with drugs like nevirapine have been incredibly effective.
And then the Berlin patient, Timothy Ray Brown, cured of HIV.
An extraordinary case. He had leukemia and HIV, got a stem cell transplant from a donor with a rare mutation in CCR5, a protein HIV uses to get into cells.
And the transplant cured both.
Astonishingly, yes.
Yeah.
Long-term remission of both leukemia and HIV off all meds. A complex, risky procedure, not easily replicated, but it showed a functional cure was possible.
Okay, so killer T cells see peptides on MHC class I. What about MHC class II? Different job.
Different job, yes. MHC class II mainly presents peptides from things the cell has taken in from the outside or from its internal recycling bins, the lysosomes.
And who recognizes those?
Primarily helper T cells. They're the conductors of the immune orchestra. They see peptides on MHC class II and then help coordinate the whole response, activating B cells, boosting killer T cells.
And dendritic cells are key presenters for MHC class II.
Absolutely. Ralph Steinman's big discovery. Dendritic cells are professional antigen presenters. Super efficient at grabbing pathogens, chopping them up, putting fragments on MHC class II and showing them to helper T cells.
They kick things off.
They really do. They also release alarm signals to activate other immune cells.
Sure.
Steinman got the Nobel for it. Phil Greenberg's tribute called him a man, a microscope, a cell, and so much more. Captures that fundamental discovery perfectly.
So this whole intricate system relies on telling self from non-self. But when that goes wrong, autoimmune disease.
Yeah, that self-non-self distinction is critical. Even simple sponges have some self-recognition. Philosophers have pondered self for ages. Medawar and Snell won a Nobel for figuring out the genetic basis, histocompatibility genes, H genes.
And when it fails?
You get autoimmunity, scleroderma, where the immune system attacks connective tissue, lupus, SLE, with that butterfly rash attacking multiple organs, the body turning on itself, horror autotoxicus, as Ehrlich called it, the horror of self-toxicity.
But could you maybe turn that toxicity against cancer, which is kind of altered self, William Coley's toxins?
That was the idea. Coley, late 19th century, injected bacterial brews into cancer patients, trying to provoke a massive immune reaction, hoping it would kill the tumor too.
Did it work?
Sometimes, maybe. Results were really inconsistent, unpredictable. And then chemotherapy and radiation came along, seemed more reliable, so Coley's toxins fell out of favor for a long time.
But the core idea using the immune system against cancer came back. Modern immunotherapy. You mentioned T-cell checkpoints, the safety switches.
Exactly. T-cells have these built-in brakes, these checkpoints, to prevent them attacking normal self-tissues. Crucial safety mechanisms.
And cancer immunotherapy often involves releasing those brakes.
There's a major strategy, yes. Drugs that block the checkpoint proteins, allowing the T-cells to attack the cancer more effectively.
And Steven Rosenberg's work with TILs, tumor-infiltrating lymphocytes, taking T-cells from the tumor.
Yeah, Rosenberg's group pioneered that. Take T-cells that have already found their way into the tumor, grow massive numbers of them in the lab, activate them, and then give them back to the patient. An army of tumor-specific T-cells.
Looking at blood again. Red cells, neutrophils, platelets, B cells, T cells. That's a whole cosmos in there, constantly working.
It really is a dynamic universe. But cancer is clever. It evolves ways to hide from that immune system, resist drugs, like creating its own protective neighborhood.
Medzetov's client cell idea. Cancer manipulating normal cells nearby.
Yeah, the idea that cancer cells can co-opt surrounding normal cells to help them survive and grow. Making the tumor microenvironment hostile to immune cells or drugs adds another layer of complexity.
And COVID-19. That was just a brutal reminder of disease at the cellular level, wasn't it? Overwhelmed hospitals, the sheer scale.
A terrible reminder. The virus hijacking our cell machinery to replicate. Initially silencing our early warning systems, like a raider getting into an unlocked house.
And then the immune system overreacting, the cytokine storm.
Exactly. In many severe cases, the immune response went haywire. Excessive inflammation causing more damage than the virus itself, a self-destructive storm. It showed how even specialized cells and organs still follow basic cell rules, but their context creates huge complexity. As Harvey said, all parts are interconnected.
And even during that chaos, other essential work, like cancer care, had to continue. The cellular battles don't stop.
No, they don't. It was a testament to healthcare workers and a reminder of the constant need to understand and fight disease at this fundamental level.
Okay, moving beyond individual cells and immunity, let's think about how they cooperate. The social cell, forming tissues, organs. It's more than just autonomy then.
Absolutely. Take the heart. Individual muscle cells coordinating perfectly, generating rhythm, transmitting electricity, contracting in sync. It acts like one single-minded entity, an orchestrated citizenship.
And neurons in the brain, we couldn't even really see them properly for ages, could we? Until Golgi's stain.
Right. Golgi's silver nitrate stain in 1873 was revolutionary. Suddenly you could see these intricate cells, the neurons. Golgi himself thought they were all physically connected, one continuous network, the reticular theory.
But Ramon y Cajal disagreed, based on the same stain.
He did. Cajal, the brilliant Spanish pathologist and artist, looked at Golgi's stains, refined the method, and drew incredibly detailed pictures. He argued forcefully that neurons were discrete cells, separate units communicating at junction synapses, the neuron doctrine.
And his drawings were the key evidence.
His drawings, his irresistible mania for drawing what he saw, were absolutely crucial. Visual proof against the continuous network idea.
And the communication is chemical, not just electrical. Loewy's frog hearts.
Loewy's 1921 experiment was beautiful. Stimulated the vagus nerve on one frog heart, took the fluid surrounding it, put it on a second heart, and the second heart slowed down too. Proved a chemical signal was released.
Acetylcholine.
Later identified as acetylcholine by Henry Dale. Convinced even the skeptics like Eccles, who initially favored purely electrical transmission.
And these neurotransmitters affect everything: mood, sleep, behavior. Paul Greengard looked at the slower effects inside the cell, linked to depression.
Yes, Greengard's Nobel-winning work showed neurotransmitters don't just act fast at the synapse, they trigger slower cascades inside the neuron, modifying proteins like DARP32. These slow pathways have lasting effects and are implicated in conditions like depression.
So depression isn't just low serotonin at the synapse, much more complex inside the cell.
Much more complex. Paul was a wonderful scientist, really dedicated to unraveling those details. I knew him a bit.
And this complexity leads to treatments like deep brain stimulation, DBS for severe depression, targeting Brodmann area 25.
Right. Helen Mayberg's pioneering work with DBS in that specific brain area, it's implicated in deep sadness. The idea is modulating activity in that circuit might help patients unresponsive to other treatments.
Delicate procedure, though.
Very delicate neurosurgery. And it highlights how little we still understand about the profound numbness some patients with treatment-resistant depression experience.
And even organs we thought were simple, like the pancreas, misunderstood for centuries. Just a cushion.
Yeah, Galen, even Vesalius thought it was mostly padding. Fallopius questioned that based on anatomy. But the big breakthrough was Banting and Best in 1921.
Removing the pancreas from dogs.
Yes. Showed it was essential for regulating blood sugar. Led to discovering insulin. Dog 410, the one that survived with insulin treatment, became famous in a way. Revealed the pancreas's vital cellular role.
And now people like Doug Melton are trying to build new insulin-producing cells from stem cells, a bioartificial pancreas, driven by his kids having type 1 diabetes.
Exactly. Doug's dedication is incredible, using stem cells to make functional beta cells. Huge potential for treating type 1 diabetes. And the kidney, too, amazing cellular sensors and transporters maintaining salt balance. It's all cellular machinery.
Just astonishing coordination everywhere. Okay, let's talk regeneration. Stem cells. Repair. You called a stem cell an oddest of great-great-great-grandfathers. Why?
Well, it captures their dual nature. Like an ancestor, they can give rise to all the different descendant cell types differentiation. But they can also make perfect copies of themselves self-renewal, like having an eternally alive twin.
The backup copy.
Right. They can sit quietly, quiescent, then wake up upon injury and divide, leading to repair. Rebirth, almost.
And the idea of stem cells in adults wasn't always accepted.
No, there was debate. But bone marrow transplantation really proved their power. E. Donald Thomas in Seattle, his pioneering work.
Huge challenges initially. Rejection.
Massive challenges. Immune rejection was the big killer early on. But gradually, with better matching immunosuppression, success rates climbed, offering cures for leukemia where none existed. Those early patient stories, Nancy and Barbara Lowry, Grushenko, they show the hope and the risks.
And then human embryonic stem cells. James Thompson, 1998. Huge excitement, huge controversy.
Yes. Thompson isolating HES cells. It opened up incredible possibilities for studying development, making any cell type. Potential for therapies was immense, but ethical concerns about using embryos were immediate and intense.
But then Yamanaka's breakthrough induced pluripotent stem cells, LPS cells, turning biological time backward.
That was the game changer, 2006. Shinya Yamanaka figured out how to take regular adult cells, skin cells, for example, and reprogram them back into a stem cell-like state using just a few key genes.
Bypassing the embryo issue.
Exactly. Suddenly you could make patient-specific stem cells without embryos. Astonishing. Reproducible. Revolutionized the field.
And your own lab works on stem cells, skeletal stem cells, OCHR cells, linked to osteoarthritis.
Yes, we've identified a population of skeletal stem cells, marked by the gremlin protein. We call them OCHRE cells. They're involved in bone growth, but also seem to play a key role in the development of osteoarthritis.
So understanding them could lead to treatments.
That's the hope. By tracking them, manipulating them, we're learning how joints develop and degenerate, trying to find ways to promote repair instead of disease.
It all comes down to that balance you mentioned. Injury versus repair, decay versus regeneration, a constant battle.
It really is a furious battle in every tissue. The rate of damage versus the rate of repair. And that balance is different everywhere. Skin repairs fast, brain tissue, not so much. That dictates how tissues age, respond to injury. Larkin's poem captures that eventual win for decay.
And the cell cycle genes, Hunt, Hartwell, Nurse's Discovery, they're controlling that repair rate, accelerators and brakes.
Absolutely. Those genes are orchestrating cell division for wound healing, maintaining tissue size, blood counts. They accelerate division when needed, put the brakes on otherwise. Essential for development, essential for repair.
And when they go wrong, cancer. Distinguishing driver mutations from passenger mutations is key.
Crucial. Cancer involves many mutations, but only some, the drivers like C and Mike, are really pushing the uncontrolled growth. Figuring out which ones matter and exactly how they work is still a major focus. Our understanding varies a lot between different mutant genes.
Which brings us back to Sam P., his melanoma. The complexity of why immunotherapy worked for some tumors but not others.
Right. His case illustrated that heterogeneity, different metastases acting like independent entities, evolving differently, responding differently, an evolutionary battle within the patient.
And ideas like cancer stem cells or reprogramming, they add more layers.
They do. The cancer stem cell model suggests a small population drives the tumor, and reprogramming concepts highlight how cancer cells hijack normal self-renewal pathways but get stuck in divide mode, locked in this selfish perpetual growth program.
But this knowledge, this ability to manipulate cells, it's leading to engineering new humans piece by piece. Artificial pancreas, liver, heart.
We're certainly moving that way. Bioartificial organs are becoming a real possibility. The artificial pancreas work by folks like Karp, Melton, Vertex, Viasite is very advanced. Bioartificial livers at Mayo Clinic, heart tissue engineering. It's happening.
Which loops back to the ethics. Michael Sandel's case against perfection. Should we eliminate unbidden gifts, enhance ourselves?
Sandel raises those deep questions. Does striving for perfection diminish our appreciation for natural gifts, for acceptance? Saletan's review mentioned violating norms like in sports or parenting. Where does therapy end and enhancement begin?
A crucial conversation for our time.
Absolutely, as the technology races ahead.
Finally, even a simple cell like a red blood cell, it holds incredible complexity inside. The song within.
Yes. The red blood cell's main job, carry oxygen via hemoglobin. But look at hemoglobin itself.
Globin proteins, heme groups, iron.
Exactly. An incredibly precise structure. 4 protein chains, 4 heme groups, 4 iron atoms right at the center. It's the iron that grabs and releases oxygen. Seems simple, but requires this intricate, perfectly designed molecular machine.
Purposeful design at the molecular level, sustaining life.
That's it, a testament to the hidden workings.
Wow, it's just staggering. The complexity, the connections we've touched on in this deep dive. From a single observation of cork to engineering organs.
It really is. Even the simplest biological units hold these astonishing mechanisms, this rich history of discovery.
And understanding these songs of the cell, as you put it, it feels absolutely essential for understanding health, disease, life itself.
Couldn't agree more. It's foundational.
So maybe a final thought for our listener, building on that song idea. Think about how all those trillions of individual cells, each playing its own intricate tune, somehow combine to create the grand symphony of a living being. You, me, everyone. What other hidden harmonies, what other secrets are still waiting to be discovered inside us?
Makes you wonder, doesn't it? There's still so much more to explore.